Hi everybody. We are going to present in the topic electronic eye for identification of tequila samples in this Congress. The collaborators were Diana Bueno, GM Gutierrez, and me, Anais Gomez. In this slide, I am going to talk about the growing interest of spirits producers to evaluate their authenticity, quality, and safety has motivated the development of efficient, portable, and low-cost analytical systems capable of monitoring these products' characteristics. The use of analytical equipment has reached a level of sophistication that allows the investigation of food's properties. Regarding spirits and liquors, a consumer commonly wants and expects a product that is pleasant to taste and smell. In this regard, tequila quality control is oriented toward understanding, characterizing, and controlling its again alcoholic content and volatile composition, which define its characteristic color, flavor, and aroma. Yeah. Tequila is the traditional Mexican liquor whose worldwide consumption ranks fourth after whiskey, vodka, and rum. The tequila is a distillate spirit from the cooked and fermented juice of the agave tequilana Weber Blue variety, whose cultivation is authorized in the Mexican states of Jalisco, Nayarit, Tamaulipas, Michoacán, and Guanajuato. Usually, tequila's quality assessment is performed using conventional analytical methods, mainly based on UV-Vis spectrophotometry, high-performance liquid chromatography, spectroscopy, chemical analysis, and sensory analysis performed by a human panel. Despite these advantages, the conventional analytical methods are long protocols that require a period from hours to days to carry out the test, expensive equipment, and require of technically qualified personnel, considering its use as complete evaluation confined to a specialized laboratory with no possibilities of adaptation in application with online quality control. Therefore, there is an urgent need for fast, cheaper, portable, and field-effective alternatives that achieve reliable and non-destructive measurements. This new perspective on analytical instrumentation has been focused on using bio-inspired systems that base their operation on the emulation of human senses to determine food characteristics such as color, shape, or size. In particular, the electronic eye has been designed to mimic human vision and analyze the color and some other attributes related to the sample's appearance, and it is usually based on computer vision, colorimetric, or spectrophotometry methods. Electronic eyes have been proved advantageous in various foodstuff areas, such as process monitoring, quality control, freshness assessment, shelf life investigation, and authenticity assessment. The main basis of electronic eyes is the acquisition of analytical information. It is shown in figure, typically using a charge-coupled device, image sensor coupled with a camera. After digital imaging, image processing is applied to enhance the acquired image for further analysis. The objective of this work is to present an ongoing research related to the development of a portable electronic eye for identification of small volumes of tequila samples by absorbance detection. In this slide, I am going to talk about experimental phase. This work focused on studying Blanco, Referencial, and Reposado tequila, and all of them were filled in UV cuvettes. Like we can see in the chart, there are two kinds of tequila, Blanco and Reposado, and tagged to identify them. Digital images were obtained with the design electronic eye system under control light using a white 2x LED and a digital camera, Raspberry Pi camera module V2, with a Sony sensor. They were controlled using a Raspberry Pi Model 3 Plus with Python 2.7 software. Samples were placed in disposable plastic cuvettes, brand UV cuvettes, free of dust and dirt to obtain trustworthy image. All digital images were processed offline to obtain RGB data using MATLAB 2018b, MathWorks, NatP, MA. In this slide, I am going to talk about all the steps that I follow in my experimental phase. The first step was the illumination of a sample with LED as light source. The next step was the image acquisition from Blanco and tequila samples. Then I had to save image in control car by Python, and all the images were shown in a display analytical information, and those images were processed by RGB, red, green, and blue. The next step was the reduction of the size of the original gray image by regions of interest. The next step was the division of pixels of each regions of interest in 3 groups: red, green, and blue. And finally, I analyzed the values in the part of surface color index and absorbance. And something important that I have to mention here is that I have 5 subsequent tests with 10 repetitions. Among those repetitions takes around 10 seconds, like you can see in the image, and these samples with the lighting is always on. In this slide, I am going to talk about the results of this experiment. The selected is automatically defined and fixed for all analyzed samples image. The ROI was chosen considering the UV viewing window of the cuvette. In this way, the region of interest and its relative position regarding the sample support is always constant. Figure shows 3 original images captured from some representative samples. The blank dotted lines within the cuvette image denote the selected ROI, as well as the RGB intensity histogram associated with it. Furthermore, it can be seen in the histogram graphs that the color components are very close to each other, especially the green and blue ones. The 3 components reach a maximum intensity value of 255 along the x-axis in graph. On the other hand, we can see the absorbance graphic and the results that I obtained in the region of interest, red, green, and blue, and all the results that I obtained and the average intensity among those regions. The blue one is the blank sample. The red one is Tequila's Blanco, and and the green one is the tequila reposado. And we can see all the difference among the three regions: the green one, the red one, and the blue one. In this slide, I am going to talk about the ensure that the internal lighting conditions don't affect the homogeneity of color in the sample. A validation of the homogeneous color distribution was carried out employing the surface color index. The change in the RGB components indicate variation of color, which are associated to the composition of the sample. The vectors R, G, and V are the combination of values required to define the result vector's magnitude in RGB space. In the case of digital image, the values of RGB are integers and range from 0 to 255, which allows generating 16777 to 16 colors, where the color white is formed with the three primary colors at their maximum value: 255, 255, and 355. It is possible to identify the two analyzed tequilas, blanco and reposado, by associating it with a ISC. Table indicates the color index data for each sample of tequila, and figure represents graphically the distribution of the 7 tequila samples analyzed. It is possible to find similarities in the figure 6 and 7. Due to absorbance and surface color index have a similar relation in the tequila samples. Less value in blanco tequilas and high values for reposado tequilas. And talking about the surface color index, we can see the blank in the part of blue color. We have the blank. In the red color, we have the tequila blanco, and in the part of green color, we have the tequila reposado. And we can see the difference among three regions: the blue one, the red one, and the green one. In this slide, I am going to talk about the discussions of this experiment. I put the discussion in several bullets, and the first one is 7 different kinds of tequila and 1 sample of distilled water as blank were tasted with the electronic eye device. 1 sole sample image were acquired by the ROI of the different samples was decomposed in its RGB components, and the number of pixels were associated to the RGB level intensities. The second bullet, the horizontal axis is divided into 3 imaginary zones, where the RGB histogram is plotted at the core. If the histogram is shifted to the left side in the horizontal axis provides information about the shadows in the image. The central zone represents the areas of the medium luminosity, and the histogram shifted to the right side indicates the light areas at 100%. The third one, the blank samples histogram, where the 3 components have an approximate value of 255. This data allowed to identify full luminosity in the electronic eye system. Fourth one, the values of the RGB components obtained were proceeded to calculate the ESC. The ISC value obtained allows validating the proposed methodology capacity by showing the possibility of discriminating different samples based on their magnitude. In this way, it was to distinguish between blanco and reposado tequila samples by their color variation. Next, mathematically, ICC value is represented as the ratio of the quotient between a factor and the root of the sum of the image RGB components. And finally, also the discrimination of the main groups of tequilas can be carried out based on the values of absorbance or the surface color index. The results encourage carrying out individual discrimination of samples using some artificial intelligence tools such as artificial neural networks or support vector machines. Both models could be fed with the color parameters obtained in order to build simple classifiers with two input parameters with no-linear mapping to the output. Future experimental stages should be considered to increase the number of analyses to strengthen the classification process and take advantage of this model's generalization capability. And finally, we have the conclusions. First, the absorbance values and the surface color index were employed as quantitative color characteristics based on the RGB model used to describe the variation of color in the sample. Second, the quantitative color characteristics based on image analysis show sensibility to detect coloration changes from apparently homogeneous samples. 3. The results suggest that image analysis employing quantitative color characteristics can identify Blanco and Reposado tequila samples. Fourth, finally, the absorbance and the surface color index show a linear trend in the absence of color. The value of absorbance and ICE is minimal and increased for color samples. And finally, using the developed electronic eye was possible to differentiate between the two kinds of tequila samples with a portable and low-cost system. And I want to acknowledge authors would like to express their gratitude to the Mexican National Council of Science and Technology, CONACYT, and author contributions. All the authors contribute equally to this work. Thanks.